Glycolysis means splitting of sugar. Glucose, which has 6 carbons, split into 2 molecules of pyruvate. There are 10 steps of glycolysis. Step 1 is the enzyme hexokinase adds a phosphate group to glucose. In the process, the phosphate group from ATP is transferred to glucose, producing glucose 6-phosphate. Step 2 is the enzyme phosphoglucoisomerase isomerize G6P into its isomer, which is fructose 6-phosphate. Step 3 is the phosphofructokinase use another ATP molecules to transfer a phosphate group to F6P in order to form fructose 1,6-biphosphate. 2 ATP molecules have been used so far. Step 4 is the enzyme aldolase splits fructose 1,6-biphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These 2 molecules are isomers of each other. Step 5 is the enzyme triose phosphate isomerase converts dihydroxyacetone phosphate into GAP. These isomers are interconverted. GAP is the substrate needed for the next step of glycolysis. Step 6 is the enzyme glyceraldehyde 3-phosphate dehydrogenase serves 2 functions. First, it dehydrogenates GAP by transferring one of its hydrogen molecules to the oxidizing agent NAD+ to form NADH plus hydrogen molecule. Next, the enzyme glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Step 7 is the enzyme phosphoglycerate kinase transfer a phosphate from bisphosphoglycerate to a molecule of ADP to form ATP. And then 3-phosphoglycerate form. Step 8 is the enzyme phosphoglycerate mutase relocates the P of the 3-phosphoglycerate from the third to the second carbon to form 2-phosphoglycerate. Step 9 is the enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenolpyruvate (PEP). The last step is the enzyme pyruvate kinase transfer a P from PEP to ADP to form ATP and pyruvate. Step 6 until the last step, which is step 10, occur twice for each glucose molecule being degraded. Therefore, 2 ATP used during glycolysis. 4 ATP and 2 NADH form, since these steps occur twice during glycolysis per glucose.
Today, we will be discussing on Krebs cycle. This is the pathway of Krebs cycle. Krebs cycle involves many enzymes and different products. Krebs cycle starts with the link reaction. In the link reaction, pyruvate will react with coenzyme A. By the help of enzyme decarboxylase, a carbon dioxide molecule is released. An acetyl-CoA will be formed with the release of a H+ ion. Pyruvate dehydrogenase will catalyze the formation of NADH from NAD+ and H+. This is an acetyl-CoA. Acetyl-CoA then reacts with oxaloacetate, involving enzyme citrate synthetase. Coenzyme A is released. Coenzyme A returns to the link reaction. Citrate is formed. Enzyme aconitase triggers citrate to change its arrangement into isocitrate. This is isocitrate. Isocitrate dehydrogenase causes the formation of NADH and H+ ion. Oxalosuccinate formed. A carbon dioxide molecule is released. Alpha-ketoglutarate is produced. With NAD+ dehydrogenase, carbon dioxide is released. Alpha-ketoglutarate reacts with coenzyme A. And NADH is formed. Succinyl-CoA is the product of reaction. With the presence of succinyl-CoA synthetase, coenzyme A is released. Succinate is formed. Enzyme succinate dehydrogenase causes FADH2 to be produced. This is fumarate. Fumarate reacts with water under the presence of fumarase. Malate is produced. Malate dehydrogenase causes the production of NADH and H+. Lastly, oxaloacetate is formed once again. This is the complete cycle of Krebs cycle, and it keeps on repeating. Energy are produced along this cycle. 3 NADH, 1 FADH2, and 1 ATP are produced in a complete cycle. 1 NADH is equivalent to 2.5 ATP. 1 FADH2 is equivalent to 1.5 ATP. Hence, 10 ATPs are produced in a cycle. However, to generate all the energies, ATPs have to be sent to the ETC.
Electron transport system, or also known as electron transport chain (ETC), is a series of compounds that transfer electrons from electron donors to electron acceptors via redox, and couples this electron transfer with the transfer of protons across a membrane in inner membrane of mitochondria. This creates an electrochemical proton gradient that drives the synthesis of adenosine triphosphate. First, electron donor will pass the electrons to more electron-negative electron acceptor. When electron acceptor accepts the electron, it will become an electron donor as it will pass the electron to the next electron acceptor. This process continues down the series until electrons are passed to oxygen, which is the most electron-negative and terminal electron acceptor in the chain. Oxygen, at the end of the series, accepts the electron and will then be reduced to water. The entire process is called oxidative phosphorylation since ADP is phosphorylated to ATP, using the energy of hydrogen oxidation in many steps. 1 NADH can generate 3 ATP, while 1 FADH2 can generate 2 ATP. ETS makes no ATP directly, and its function is to ease the flow of electrons from food to oxygen.